All right, so this is kind of where we left off. We were talking about how PFK1 and FBPase1 activity is regulated in liver cells by the two hormones, glucagon and insulin, right? So, right, we said PFK1 was our glycolytic enzyme, FBPase1 is our gluconeogenic enzyme. And so what do insulin and glucagon actually affect in the cell? And it's the levels of fructose 2,6 bisphosphate. So that's how we translate the signal from being extracellular to being intracellular. And we said that structurally, that FBP, the fructose 2,6 bisphosphate, is really similar in structure to fructose 6-phosphate and fructose 1,6 bisphosphate. But here's the difference. The fructose 2,6 is not a metabolic intermediate in either glycolysis or gluconeogenesis. So it's only used in regulation. It is not used in actual metabolism. So if you have lots of this molecule of fructose 2,6 bisphosphate floating around, it is going to increase PFK1's affinity for fructose 6-phosphate. And I mean, it increases it 25-fold. So that's a lot. So you got to roll through glycolysis and do lots of glycolysis. But at the same time, those high levels of fructose 2,6 bisphosphate really change the affinity of FBPase1 for its substrate, which is fructose 1,6 bisphosphate, and that affinity goes down. So if the FBPase1 can't bind to its substrate, you're going to inhibit gluconeogenesis. So when one goes up, the other goes down. So what regulates these levels? We said those were our two hormones, insulin and glucagon. And what kind of signals are you getting? If you're producing insulin, that means you have really high, well, relatively high blood glucose levels. So you're going to make more fructose 2,6 bisphosphate. Glucagon, on the other hand, shows that, well, signals that you have really low glucose levels, blood glucose. So you haven't eaten in a while. And so it's going to decrease the levels of that fructose 2,6 bisphosphate. But how? And so this is the control of that dual functioning enzyme that was up here in the picture. This and this are the same thing. So just two different states of the same enzyme. So this enzyme has two names because it kind of has two functions. It's called phosphofructokinase 2 and FBPase2. So each of the two parts of this enzyme have their own unique functions. So the first part is the kinase domain, right? And so the kinase domain is the PFK2 domain, and that one will phosphorylate fructose 6-phosphate into fructose 2,6 bisphosphate. The other half of the molecule, the enzyme, is a phosphatase, and that's the FBPase2. And this will dephosphorylate, so remove the phosphate group from 2,6 bisphosphate. So you're going to lose the phosphate on the second carbon, and you're going to turn into fructose 6-phosphate. So then you get the question of, well, how is this enzyme actually regulated? We said it's insulin and glucagon, but how do insulin and glucagon actually affect this enzyme? And it's through phosphorylation. So this enzyme can basically have two states. So let me copy this. So you can have a state where it is unphosphorylated, where you have a serine residue that has a hydroxyl on it. This is the unphosphorylated, or dephosphorylated, however you want to say that. Or that same serine residue could be phosphorylated, right? And so there's only one site on the enzyme that needs to be phosphorylated. If I could go back. So this is phosphorylated. And so the activity of the enzyme is going to be controlled by the enzyme's phosphorylation status. So if you have high blood glucose, you are making lots of insulin. And so that dual functioning enzyme is going to be dephosphorylated. PFK2, FBPase2 is dephosphorylated. if the enzyme is dephosphorylated, you kind of have two things going on. Number one, PFK2 activity is increased. So you see a great increase in PFK2 activity. When PFK2 is active, what happens? It's a kinase, so it's going to convert that fructose 6-phosphate into fructose 2,6-bisphosphate. And we know that fructose 2,6-bisphosphate is a positive regulator of PFK1. So now PFK1, right, is going to be positively regulated, right? It's going to make it very active. So now it has this really high affinity for its substrate of fructose 6-phosphate, which is then converted to fructose 1,6-bisphosphate. So you are cranking out, going through glycolysis, lots and lots of glycolysis. But remember, at the same time, because you made lots of fructose 6-bisphosphate, that is going to be a negative regulator of FBPase1. And so it's going to prevent that conversion of the fructose 1,6-bisphosphate to fructose 6-phosphate. So we are going to inhibit gluconeogenesis. It's going to go down a lot. So remember that if you're going to promote one, in terms of like glycolysis and gluconeogenesis, if you promote glycolysis, you're going to want to inhibit gluconeogenesis. So always remember that's a twofold thing. All right, so here's our other condition where we have low blood glucose. Oh, and I think I forgot to say, but I wrote it, but I forgot to say. So when you have high blood glucose, you're going to make insulin. What does insulin do? Insulin activates protein phosphatase 1, which we remember from last semester, and protein phosphatase dephosphorylates, right? So you're left with that hydroxyl. Okay, sorry. All right, so in our second condition, we have low blood glucose. So we are going to have glucagon floating around in our bloodstream. Glucagon, when it enters the cell, is going to activate protein kinase A, pKa. We remember pKa. pKa, one of pKa's substrates, right? pKa has lots of substrates, is the PFK2 portion of this molecule. So this dual functioning enzyme is going to now be phosphorylated. If it's phosphorylated, then that means that FBPase2 activity will be high. So now what does FBPase2 do? It removes a phosphate from fructose 2,6-bisphosphate, turns it into fructose 6-phosphate. At the same time, it's also going to decrease PFK1's affinity for fructose 6-phosphate, right? And you're also going to increase our FBPase1's affinity for fructose 1,6-bisphosphate conversion to fructose 6-phosphate. So at the same time that we're decreasing glycolysis, we're also going to increase gluconeogenesis, because once you get fructose 6-phosphate, then it can go through gluconeogenesis and we can eventually make glucose, which is kind of the name of the game there. All right, so let's kind of look at our picture now that we know sort of what's happening here. Depending on what's happening, let's say you haven't eaten, right? So you have low blood sugar. You're going to make glucagon. Glucagon comes into the cell, activates protein kinase A. Protein kinase A converts this dual functioning molecule from being dephosphorylated to being phosphorylated. So this is all in the presence of glucagon. Once it is phosphorylated, now this active enzyme is going to have an active FBPase2 domain, which means that it's going to convert fructose 2,6-bisphosphate into fructose 6-phosphate. You just kind of have to go through this a couple of times with yourself and look at what's the difference on if I have high blood sugar versus low blood sugar, and kind of track through what gets dephosphorylated, what gets phosphorylated. And if you look in these, sometimes biochemists will do this. They'll put an asterisk next to the part of the molecule that is active. So in the presence, when we did glucagon over here, right, this dual functioning enzyme became phosphorylated. So here's the phosphorylated state. So in the phosphorylated state, what part is active? Oh, well, that's the FBPase2, and that's why it has an asterisk. On the other hand, if it is dephosphorylated, when you have high blood sugar, right, there's no phosphate there, it's just OH. Now the PFK2 portion is active. So, okay. So now we got to talk a little bit about something else, right? Remember from chapter 13, the storage form of glucose is glycogen. And glycogen, if you remember, right, is branched glucose, right? Glucose homo polymer, right? With what kind of linkages? It had alpha 1, 4. This is the chain, right? CHA, chain, the straight chain. And then it had alpha 1, 6 branch glycosidic bonds, right? So you got to remember that about your different sugars. That's important. Okay, so what is glycogen? I hope you remember in class, I had the Kush ball, right? And it has this little core, and then it has all these little, things that come out of it. And it's really similar to glycogen. The only thing that a Kush ball doesn't model is that on every single one of these glycogen chains, you're going to have branches, right? And branches can come off of there, and branches can come off of there, right? So it's very, very, very highly branched, and so that's kind of the difference between the two. But if you look at that glycogen in that core, in this red center of a core, there's actually a protein, and that's called glycogenin. And so that protein is what allows the sugars to attach and kind of keeps everybody together. So in one glycogen core complex, you have about 50,000 glucose molecules, right? And so there are straight chains, and then on those straight chains, you will get a branch every about 8 to 12 residues, which means that in this 50,000 glucose molecules, you have about 2,000 non-reducing ends, which are ready to be cut off and shipped to the bloodstream lickety split as quick as you can, right? And so if you look like under a microscope at glycogen containing cells, what you'll see is that there are actually 20 to 40 glycogen core complexes that form one microscopic glycogen particle. So if you really think about how much glycogen is available in this super, super quick time frame, it's a lot. It's a lot. So we know that we have glycogen in two places. We have it in the muscles and we have it in the liver. And so if you think about the function of the muscle and the liver, if you have glycogen store in the muscle, do you really want to ship out that glucose to another cell type? And the answer is no. You want to trap the glucose inside the muscle cell. So you stored glycogen in the muscle. You want to use the glucose in the muscle. You do not want to ship it out and feed somebody else. So the end product of removing glucose from glycogen in muscles is glucose 6-phosphate, because now it is still trapped in the muscle cell. Because muscle cells need so much energy, they don't want to lose the whatever glycogen stores they had. So it sustains really vigorous exercise for about an hour or so, which is pretty good. All right, then you also have glucose in your liver cells. Now this is really for export, right? Your brain needs it. Whatever needs it, the liver is going to send it. So this is, you could say like this is between meals. How do you get energy to different parts of your body? And that is the glycogen storage. So it's about 10% of your liver. In your muscle, it's only about, 1 to 2% of your muscle. So you have much more in your liver, but you have more muscles all around you. Okay, hold on, y'all. I got to pause for a second. Let's see. Okay. All right. So let's talk about how that glycogen is regulated. We know that making and breaking down glycogen occurs in the cytosol, right? The substrates for the reaction are really the ends of those branched polymers, right? The non-reducing ends, the free ends, where the anomeric carbon is not in a glycosidic bond. And so it's regulated really by three enzymes: glycogen phosphorylase. And I'm going to preface this, I'm going to tell you a little bit more than kind of the book tells you right now. But in order for glycogen phosphorylase to be active, this enzyme must be phosphorylated. So the enzyme plus phosphorylation, we're going to say equals activation, right? Because phosphorylation status controls so many on and off switches, right? So the enzymes are always present, but whether or not they are phosphorylated is controlled by cellular conditions, which will then lead to its either activation or deactivation. Glycogen synthase, if this one is phosphorylated, so if the enzyme is phosphorylated, then it is active. Oh, it is, I'm sorry, it is inactivated. We got to do the opposite, right? Phosphorylase and synthase, right? So synthase is inhibited by phosphorylation. And then you have glycogen branching and debranching enzymes. These are going to be regulated a little bit different, and we're not going to get into as much detail as we are glycogen phosphorylase and glycogen synthase. So these, how are these regulated? How do we know they become phosphorylated and dephosphorylated? And this is all about all those hormones, right? Epinephrine and insulin and glucagon, right? Those are all the ones that control the phosphorylation and dephosphorylation, so plus and minus phosphorylation, that lead to changes in the enzyme's activity. But the enzymes are also regulated. So all of these, so this is really regulation. So they're regulated by that phosphorylation, dephosphorylation, but they're also regulated by allosteric control. ALL, allosteric control, right? So, and when we talk about allosteric control, like that's something binding and changing the conformation of the protein. So a lot of times this has to do with the metabolic state of the cell. So like the EC of the cell, the charge of the cell. So when you're thinking about these two big ones, right? How are they regulated? And it's kind of twofold, right? Okay. So when we talk about glycogen, you kind of got to remember the general things in how these glycogen molecules are made, right? And so I don't think they're labeled on this diagram, but you remember that the chain linkage, right, is always going to be an alpha 1,4, and then a branch linkage where you're going to go up this way. That's a branch linkage. That's an alpha 1,6. Okay, so just kind of keep that in mind. Okay, I'm going to erase that because I'm going to end up drawing all over this. Okay, so we need all of these branching enzymes to make all of those free ends so that when that bear walks into the room, remember, we can get a boost of glucose in our bloodstream and run out. So the regulation of glycogen being either made or used depends on three key reactions. So the first key reaction here is glycogen phosphorylase. Glycogen phosphorylase is what is going to release our glucose 1-phosphate molecule. So we're going to take something that's attached to a chain, right? It's attached to a straight chain, and we're going to cut the glycosidic bond, right? And we are going to cut it right there, and then we're going to add a phosphate group to it. And so now glycogen phosphorylase, we're going to end up having, we'll take our inorganic phosphate, right? And we're going to add it to our molecule, and we're going to get a glucose 1-phosphate. Now, glucose 1-phosphate is not something that can be dephosphorylated, right? Because you're kind of thinking either we want it for the muscle, and then in that case, we still need glucose 6-phosphate, or we need to be able to ship it out to places like the brain, right? Either way, if you want to even be able to dephosphorylate it, you're going to have to convert from glucose 1-phosphate all the way over to glucose 6-phosphate. So this is just a mutase that will change the position of the phosphate group into the glucose 6 phosphate. And then depending on what cell type you're in is what's going to happen. So how are we going to activate glycogen phosphorylase? What hormones are going to do that? And those hormones are glucagon, and epinephrine. Okay. The second really important reaction is the reaction of glycogen synthase. So this is the opposite. Glycogen phosphorylase and glycogen synthase, these two are opposites, right? So remember, if we're going to promote one, we're going to inhibit the other. So glycogen synthase is activated by insulin. So when we eat a lot, right? If we have excess sugar in the blood, then we're going to use it to store it as glycogen first. And then if our glycogen stores end up full, then we'll have, fat being made and things like that. Okay. So glycogen synthase, activated by insulin. It's going to add a glucose, but not just any old glucose. It's going to use a uridine diphosphate glucose, a UDP glucose, and it's going to add that to a non-reducing end. And when it adds that to our non-reducing end, that's a free end for the carbon to be adding. Then what we can do is we're going to lose the UDP. Then the UDP is converted from UDP into UTP with the use of an ATP molecule, right? So this is costing us. To do this is costing us energy. We can regenerate it with another free glucose. Well, it's not quite free. It's really a glucose 1 phosphate. And we can add that to our new UTP and we make a new UDP glucose. Okay. So anytime we're going to add a glucose molecule to the glycogen ball that's already there, it's going to cost us an ATP molecule. But you got to think about, where did that glucose molecule come from the start? Well, really, it came from the blood. And so in its transition from the blood into the cell, glucose is not phosphorylated, but to trap it, we have to phosphorylate it. So that happens through the same enzyme, hexokinase. And so you really actually have an investment of 2 ATP, because you have to invest 1 ATP in order to trap the glucose in the cell. Then you're going to go in, and when the UDP is regenerated, you're going to use up another ATP molecule. So it is going to cost you some ATP to be doing this, but theoretically, if your blood sugar's high, you've got plenty of energy, and that's not a big deal. Okay. And the third reaction is this debranching enzyme or branching enzyme. So we're kind of grouping them together. And so these enzymes make sure that glycogen, phosphorylase, and synthase have access to really the maximum number of those non-reducing ends for either cleavage or formation, right? So we can very quickly make up our glycogen stores. So let's say you use up your glycogen, you haven't eaten in a while, whatever it is. As soon as you eat and you get a high enough blood sugar, you're going to start remaking that glycogen, and you make it really, really fast. Okay. And so these are those alpha 1,4. So these are the alpha. They start with something that's on alpha 1,4 glycosidic bonds, and then they are going to make the alpha 1,6, right? Okay. So let's get into the nitty gritty of how all this works. So glycogen degradation. If you are using your muscles, you have a relatively low amount of ATP available and ready to go for those muscles to work. So as soon as you're exercising for an extended period of time, you're going to deplete the cytosolic levels of ATP, and you're going to have to resort to using glycogen. Okay. This conversion of going to use glycogen is initiated by our enzyme, glycogen phosphorylase. Glycogen phosphorylase is a homodimer protein, right? And remember, we said if it's phosphorylated, if we have that phosphorylation group, then it is active. ACTIV, it's active, right? If you dephosphorylate it, then it is inactive. Okay, so just kind of keep that in mind. So this one is going to exist in those two conformations based on its phosphorylation state. So if it's phosphorylated, it's active and it's R. If it's dephosphorylated, it is inactive and it's T. So remember the relaxed, intense state of hemoglobin? It's the same sort of deal. It is going to catalyze the cleavage of those alpha-1,4 glycosidic bonds at non-reducing ends. So it actually, it's actually kind of cool. It stays attached. The enzyme stays attached to the glycogen. And then it kind of just like goes along the glycogen molecule and keeps cleaving and keeps cleaving and keeps cleaving and keeps cleaving until it gets within 4 glucose units of a branch point. And then when it gets to that branch point, 4 units away, it can't get any closer. So you have to have a debranching enzyme come in, cut off the branch, then it can keep going. Okay, so here's what we said. We said it's a dimer, right? And we said we have 2 states. We have the T state and we have the R state. So let's see what happens depending on our hormone levels. So if you have glucagon or if you have epinephrine. Now glucagon is specific to the liver. Epinephrine is ubiquitous for both liver and muscle, right? And so if you have the presence of either one of these hormones in your bloodstream, it either means you haven't eaten in a while or the bear's coming in, but regardless, you need to pump some glucose into your bloodstream. So it is going to activate phosphorylase kinase. It is going to activate the kinase so that the enzyme will convert from the dephosphorylated into the phosphorylated state. The phosphorylated state is the active state. And so this is where we're going to go. And if you have this, you're going to go and you're going to build glycogen because my enzyme is active and that's great, right? Did I go the wrong way? You're not going to build glycogen. I went the right way. I just said the wrong thing. You're not going to build glycogen. You're going to go this way, right? You're going to phosphorylate it. And this is glycogen phosphorylase. You're not going to build glycogen. You are going to degrade glycogen. It's like, what? Degrade glycogen, right? Because this enzyme is active. Now you're going to go and you're going to degrade your glycogen so that you can go and increase your blood sugar. Now, on the other hand, what happens if you haven't eaten in a while? You have eaten, right? You've eaten that bagel. It spiked your blood sugar. So your pancreas is going to make insulin. Your insulin is going to go in the bloodstream. It's going to get to your liver, and it's going to activate protein phosphatase 1. So protein phosphatase 1 is going to go in the opposite direction because it's going to dephosphorylate the enzyme. So now we have these serine residues that do not have phosphorate groups attached to them, right? And so in this state, now you're going to have an inactive enzyme. So if it's an inactive enzyme, you're not going to degrade your glycogen, right? This is an enzyme that only has one function. So glycogen phosphorylase, what is its job? Its job is to degrade, degrade glycogen. So if it's on in the active state, it's on, it's going to degrade. If it's in the inactive state, then it's not going to degrade. So it's kind of that phosphorylation is that on-off switch, and it's going to tell you whether something is active or not. So then if you look on the opposite side and we look at glycogen phosphorylase kinase, right? So now we have something that is going to add a phosphate group. So this is really a key in our epinephrine signaling. And let's see. So if we look in muscle cells, right? If we look in muscle cells, if you have epinephrine is telling you that the bear's coming into the room, right? Whatever it is. So we're going to activate phosphorylase kinase. We're going to take that and we are going to phosphorylate our enzyme. And so now we're in the R state. We're active. We're phosphorylated. But there's another level to this regulation. If you really want this to be very active, you also are dependent upon the energy charge of the cell. And so ADP, ATP is going to have an effect on this. So if you have lots of AMP, it's actually going to bind to this enzyme and turn it into an active but unphosphorylated state, right? So it's going to actually bind here and it's going to inhibit its activity. So the most active form of our enzyme, right, is right here. It's phosphorylated and it is not bound to AMP. Because usually you're not going to really be building glycogen unless you have enough ATP to actually do it. So the other thing, so ATP, it's not really ATP, it's really AMP. And so if you look at the binding site of where AMP binds, there are actually two different things that can bind at that same site. You can have AMP binding, or you can have glucose. This is, and it's just a little circle, but it's glucose 6-phosphate, right? Or ATP could be there, but more often than not, it's glucose 6-phosphate. So the AMP is going to compete with really that glucose 6-phosphate for binding. So if you have something bound there, if you have glucose 6-phosphate, if you have ATP bound there, it's going to affect the activity of the protein.